going? It's busy, you know. It's very close to the end of the semester and getting crazy now.
Yeah.
Lots of grading, lots of, I need to put tests, I need to put practical, and need to respond to emails from students. So it's
Hmm.
kind of busy, but you know, it's almost there, almost done.
Sure.
It will
And
be a
then
busy week for everybody, as a matter of fact.
doesn't fall semester start soon?
Fall semester is going to start, I believe, August. I believe it is August, very close to the end of August.
Okay.
The last week of August. Yeah,
So you'll have maybe 2-3 weeks
almost.
before the
Yeah,
next.
like 2 weeks, because you know we have, after we finish with the finals and stuff like that.
Uh-huh.
We have 1 week, you know, to post grades and respond, you know, to students' email and stuff like that. So it will be next week will be a busy week for this purpose,
Hmm.
and then we'll have maybe 10 days break before we hit the fall semester again.
And then is the fall semester going to look similar to the summer?
Yeah, it is going to be, till now they decided to have it also like virtual, and then we are going to have, like, very close to the end of the semester, we're going to have like some face-to-face sessions.
Mm.
Yeah, so, you know, and I'm not sure about that, because they told us to have like a face-to-face for the summer, like
Uh-huh.
the last 2 weeks, week 9 and 10. And most of the students did, you know, disagreed. They refused to, you know, like, be face to face, you know, under these circumstances, which I agree with them. Cases are like increasing every day,
Hmm.
and that's exactly, doesn't make sense to have this kind of meeting at this time.
Yeah.
Yeah, especially for the lab, they mentioned, you know, we have to maybe arrange face to face for the lab. The lab itself, you have to have 1 week for safety procedure. So you need to talk about safety and what you should do in a microbiology lab setting. That is 1 week. And then what kind of experiment you can do in 1 day and be able to see the results, you know.
Yeah.
So it didn't work. It didn't work. So they decided to, you know, postpone it to the fall semester and probably give it a little bit more time, not just 2 weeks, maybe extended it a little bit more.
Yeah.
We'll see. We'll see.
Well, for me, just as a student, um, I get so much more out of being able to listen to a lecture and
Yes, everybody
actually
does.
hands-on.
I agree
Yeah.
with you. Everybody does. Everybody does. Do you think it's easy, you know, to have like virtual lab or Zoom meetings? You know, it is not the same like when you have face-to-face lecture or labs. You have, you know, you can speak to your instructor. The instructor can
Hmm.
speak to you. You can, you know, have more detailed discussion and stuff like that. Definitely, especially for science lab, science lectures and subjects, biology, microbiology, anatomy and physiology, biochemistry, all those, you know, really need face-to-face
Yeah.
instruction. I agree with that. Yeah.
And especially, I was reviewing the chapters. Which one was it? I'm flipping through. Just a moment, please. Which one was it? I think it was the pathogenic DNA viruses. And as it started getting into really details, about 3/4 of the way um, through the chapter handout provided,
Yeah.
I mean, it just kind of lost me. I mean, because it was just,
Too much materials, you mean?
well, it was just because there's no context. It's like I don't have context on all the
Do you have
verbiage.
the textbook?
No, I don't.
Yeah, this would be a problem because the textbook is so important, because the textbook is going to be your backup for these kind of presentations.
Mm-hmm.
Yeah, and I can work a little bit more for this. This is chapter 20, 20
I think it is.
24.
I'm trying to find exactly where my eyes just kind of glazed over, and it's like, what am I reading?
It's like a
Um,
virus because yeah. Let me start. Take it from here.
it might even
And
have been the adaptive immunity.
oh, you
Um,
mean the immunity, not the virus?
I think.
Oh, you
Uh,
mean those like clonal expansion and stuff like that?
yes, the cytotoxic T cells, clonal deletion of T cells.
Uh-huh.
I think that's. I'm sorry, I'm flipping through. It was just earlier, and
Yeah, that's okay.
I,
Yeah, that's fine. Let me go ahead and just need to go through some important
yeah,
points here, and then we can see this together.
yeah,
So first of all, you know that this week is going to be your final. Okay, so you have two things going on. One of them is the final practical, and that is your second lab practical. Will be exactly similar to the
mm-hmm.
first one that you have 1 month ago. That is going to be available for you starting Tuesday, uh, August 4, and it will last for like 2 days. So it will finish August 5th,
Yeah,
midnight. Okay.
yeah, Professor, on that. And you may be about to say this, but can I just to understand? You had listed out, and I had asked you, could you list out the different lab handouts we should be focusing on, just to verify? And you did list out one, 4, 5, 6, 7, 8, like 9 or 10, all the biochemical testing unknowns.
Yeah,
There's
so
5.
basically, we are going to cover, practical is going to cover everything we had after, we have after practical one, okay?
Okay.
And that includes the identification of the unknown gram-negative bacteria, Enterobacteriaceae, the different genera that we mentioned.
Yeah.
Also include the unknown Staphylococcus and Streptococcus. What I mean by that, I'm not going to give you like isolate like what I did for your assignment and give you lots of information and ask you to interpret. You already have done that. What you need to do is basically to study for all the biochemical testing. You need to study to know the different kind of media used, different kind of indicators used, and the purpose of those indicators. If there is a biochemical test that need to be visualized or interpreted by adding a reagent, a specific reagent, you need to know the name of the reagents. And then finally, you need to be able, if I give you a test tube with a specific culture and say, ask, is this like a bacterium was capable of fermenting lactose or no, for example.
Mm-hmm.
So you need to look at the picture, and based on the color that you will see, you need to tell me. And then I may follow up and ask you what kind of pH, what is the name of the pH indicator used. You need to tell me like this is phenol red, for example, right? So, something like this. So all the biochemical tests that were covered under Enterobacteriaceae and Staphylococcus and Streptococcus labs, you need to be really know those. Okay.
Mm-hmm.
You have a lab which was kind of a follow-up the urinary tract culture.
Mm-hmm.
Okay, again, different kind of media was used, so we need to know the name of those media, and what kind of indicator was used, and based on the colony shape, for example, I will ask you, based on the shape on the plate for urinary tract culture, you need to tell me what you think the genus is, something like that.
Mm-hmm.
Okay, so those for those labs. Then you have other labs like immunology and serology. You need to know the difference between antigen and antibodies. Those three different tests that were mentioned in the lab assignments. You need to know each one of those and what you are detecting. Are you detecting antigen? Are you detecting antibody? Stuff like that. That will be a kind of an easy lab. Then the one that also need your attention will be the bacteriophage lab. And again, you should be capable of, if I give you like a question or like a diagram or a figure about how to calculate the phage forming unit or plaque forming unit. You need to know what is PFU stands for, plaque forming unit. And you need to know also how to be able to calculate based on some of the information that I will give you. You need to follow exactly the same kind of formula that you used for colony forming unit or CFU per mil. Same formula exactly, but instead of counting colonies, you are going to count plaque. And those plaque is basically, you know, that those are clear zone on the agar. And why it is clear? Because those are dead bacterial cells that already got infected with the bacteriophage and lysed. So they are not exist, okay? And instead you will see like a hollow in the media. And that hollow is basically filled with bacteriophage, okay? So you need to be capable of taking the information, counting the number of plaque forming unit in the plate. You need to know the dilution. You need to know amount plated, and then you can use the formula, same formula for colony forming unit to be able to get the number. This is important as a matter of fact, okay? I will go through this in detail. So whatever lab you have some question, we can do it together. Then you have two labs, the ectoparasite, and those ectoparasites, you need to know the difference between arachnida, class arachnida, and insecta, how to be able to differentiate, the main feature for differentiation between the two. And all those, you'll be able to see if I give you like a picture of a louse, you need to tell me this is a louse. You know, if I ask you about the specific name or the scientific name, you need to memorize those names. Ticks, you need to know the difference between ticks and mites, all these kind of things. Okay, and finally, you have the helminths. Helminths also need to know the difference between the two different phyla, nematode and platyhelminthes. You need to know this, how to differentiate between those. Looking at those worm that I have in the presentation, there are names, general names, and there are scientific names. You need to know both. So if I ask you, give you a picture, and ask you to give me the general name, or the non-scientific name for that particular organism, you need to be able to do that. Or if I ask you to give me the genus name, you also need to be able to do that. That will leave only one lab, the API, and that will be an easy. You need to be able to interpret numbering. If I give you like a chart for an API, plus minus, and ask you to interpret the numbers, you need to give me the numbers. Exactly similar to the lab assignments. Those are mainly the labs that are going to be covered. And let me tell you, it will be, okay, this week, you have a lab practical and you have a final. Those are going to be kind of tough a little bit, okay? Yeah. So they are going to be tough. So expect to see some kind of toughness a little bit. So we really need to be prepared for these two, okay? This is why I let them at the end, so you give you more time to study for those. So again, look at those all biochemical features for gram-negative, for staph and strep, and be able to interpret the data from a figure that I'm going to provide you with. Media, you need to know the name. If I give you, I said, what is CNA stands for? Okay,
Mhm.
you need to tell me what CNA stands for. And if I ask you what kind of, is it like what kind of media? Is it selective? Is it differential or both? Okay, what kind of, like, what is the selective pressure that was added to the media? You need to know this also. Okay, what else? The lab practical, because the department agreed that it will be a no backtrack. So you will have one question at a time. So you will take your time basically to answer the question. There is no need to leave it and go, because you are not going to be come back to that one, because they need, have some restrictions, in this kind of test.
Yeah, and that was really a time management
Yeah, I'm going to
process.
increase the time a little bit. I know that the first practical I gave like 50 minutes. I believe it was around 50, 60 minutes. So I'm going to extend the time a little bit more, so it can give you more chance to think. Because some of the question multiple choice, easy, some of them true or false, and some will be calculation. So like, for example, if I give you like 1.5 minutes per question, some question will take 10 seconds, right?
Yeah.
And some will take like maybe 2 minutes. So the remaining time from the 1.5 minutes that you already spent 10 seconds for, you will add it to the 1.5 minutes for that particular one. So you will have like very close to 3 minutes. So you have extra time to be able to solve it. Okay.
Yeah.
So yeah, it is important. I'm emphasizing on this, please. That practical is going to be kind of a little bit harder than the first one. And the final test also is going to be a little bit harder. If you look at the screen here that I'm sharing with, this is the grading policy that I sent a few days ago. So you can see, can you see the screen?
Yes.
Okay, so this is the lab part, and the lab part is around 38% from the total grade. The lecture is going to be approximately 62%. Okay, remember, if you look at your grade book, some of the assignments I didn't grade it yet, but I promise, as soon as I post the lab practical and the final, that will give me some room to start grading all those lab assignment left. And I think the presentation, I didn't touch it yet, the PowerPoint presentation, the 50 points for the presentation, I didn't touch yet, so I need to do this. What else? The final is going to be cumulative. It's only 100 points, not 150, 100 points.
Mm-hmm.
Okay, and I made it kind of easy by minimizing. I don't need from you to go back all the way to chapter number 1. That will be ridiculous, especially the timing for the summer semester is very short and very tight. So this is why for the final I selected 2 older chapters that I believe are important and somehow connected, chapter 6 and chapter 10. Chapter 6 is the metabolism, and chapter 10 is the antimicrobial agents using like antibiotics generally. Okay.
Yeah.
So
And we've
those
done
are
those before, right, Petra? I mean, we've done 6 and 10 already, haven't we?
yes. I mean, because the policy is going to, that the final should be cumulative, meaning whatever left,
Yeah.
and then plus some previous chapters.
Okay.
So I selected only 2 chapters for you.
Yeah.
Okay. I didn't select the others. The others like chapter 1, 2, 3, 4, 5, and 7, 9, all these kind of things, I excluded those completely. So to make you focus more on the new chapters, and new chapters, as you know, 2 of those are immunology, 15 and 16. One is talking about the innate immunity, and the second one is talking about the adaptive immunity. Those are 2 chapters connected. And then you have 2 chapters for viruses, RNA viruses and DNA viruses.
Mm-hmm.
Okay. And here you need for those particular chapters, 15, 16, 24, and 25, don't solely depend on the lecture. You need to read and fill the gaps from the textbook. Okay, it is important. Okay, so you need to really study for whatever gaps you can see or you can see in the PowerPoint presentation. If there's some gaps, you need to fill these gaps by coming back to the textbook. Okay, so this is also important point for this Zoom meeting. So what else? I have like the lab starting Tuesday, finishing Wednesday, and the final test starting Wednesday and finish Thursday. Because Thursday, August 6th, is officially the last day of the summer semester. Okay, so you have 1 day overlap between the two. So if you start, like you finish your practical Tuesday, I will give you more time to study for the final and make the final Thursday, for example.
Yeah.
Okay. Those together are 200 points. So lab practical is 100 point. The final is 100 point. So like 20% roughly, because I can see if you add 380 points plus 621, so you are talking about here 1,000, a little bit more. So those 200 is 20% of your total grade. The homework, I didn't post the grade yet. As I mentioned before, as soon as everybody will finish all those assignments, then I will extract your grades and put it in the grade book for you. They count for 99.5 points. Okay. So any question for me for the grading policy and for what is going to be covered for the final lab and the final lecture exam?
I have a question. For one of the homework assignments, it was due on the day that we had one of our exams.
Yes.
I emailed you. I did it like the following morning, and you just told me to like remind you because it says late, 0 points on the mastering homework, like the score page.
Yes.
Like, do I get any credit for that, or should I just like?
Yes, I will. As I mentioned, I will go all the mastering homework and see whatever you collected. I will post it for sure.
Okay.
Yeah.
All right. I was like sweating that one. Okay.
No, you don't need to worry about this. Yeah. Yeah. Yes. Okay. So for the lab now, you have any kind of specific lab that you really, you have something which is kind of confusing, and you need to, have more information or understand more for any lab?
Carol? Is this Carol, the other student?
I believe yes.
Yes.
Carol?
Yes.
Okay, I think Carol and I have the same question.
Yes. For what?
We went back and forth a little bit on our tech staff that we had, but on the PFU calculations, and I know we've done it with CFU, and I think I'm leaning towards Carol is right, but on the calculation.
Yeah, let me share with you this.
I don't expect you. Could you bring up question 3?
Okay, question number 3.
And it was on. I'm sorry, it was on the bacteriophage.
Yeah. Yes.
Yeah.
I'm sharing this presentation with you right now.
Yeah, we see it. For the denominator, so for instance, on the T4R, I chose the 125 colony because it's between 30 and 300.
Perfect.
And then it's it's 10 to the negative 6 dilution. Yes. So as a denominator, the way I did the math was 125 over and I'm not sure. Carol had 0.1 and I had just 1.
Oh, you mean the amount plated?
Yeah.
The amount plated is 0.1 because look
0.1.
here. Okay, so look here.
Uh-huh.
So this is your viral specimen, right?
Yeah.
And you take 1 mil from this and you started all the good stuff by making all this serial dilution here, right?
Mm-hmm.
So if you look, the actual virus or the actual phage that you was plating or you counting is basically the 0.1 mil.
Got it. Okay.
This is the 0.1 mil that you are taking here, okay? Because you add those 0.1 mil to other stuff, and those other stuff is agar and soft agar that you need to mix and then pour everything over the, you need to do overlay over the plates. This is the way when you do the phage assay, okay? So you make your dilution from the phage, you mix it. You don't need to, it is not exactly the same like colony forming unit where you take straight from the dilution and streak over plates. Basically, you mix it with the soft agar and then pour everything, to the plate here, okay? Or even like if you do this, so you need to look for the number here that this is the real number that you were counting, the 0.1 mil,
Yeah. Okay.
okay?
So
Here, don't get confused. I know that maybe you got confused because it says here 0.3 mil E. coli. Here, the whole thing here is basically you are mixing 0.1 mil phage plus 0.3 mil of your E. coli, okay? Because you need to mix those first. You need to get a chance for the phage to infect the E. coli. You incubate those together like for 5 to 10 minutes, and then mix everything with 3 ml soft agar, and then pour it over the plates. So actually you are counting 0.1 ml of the phage. This is what you are after,
Mm-hmm.
okay? So again, you mix the phage plus the E. coli culture here, 0.3 ml E. coli plus 0.1 ml phage. That will make it 0.4. You let this incubate for like few minutes at 37. So you give a chance for the virus to attach and start the process, attachment, entry, and all the good stuff about synthesis and packaging, stuff like this. And then you mix it with the soft agar and pour everything, and then allow this to solidify, incubate it for 24 hours, 48 hours. Then you look for plaque. And as you mentioned, this is the right number that you are after. Like here, you said the 125 because it is between 50 and 300. It is exactly like the colony forming unit. Yes.
Mm-hmm.
Yeah.
Okay.
Okay.
Right.
So
Yeah.
this is to explain. So expect to see maybe a couple of questions about this calculation. So you need to be able to to know exactly how much you are plated, what was the dilution factor that you selected, how many plaque forming unit you are counting, and then do your calculations. This is for the phage, yes.
yeah. Okay, I have a question, and this is probably a dumb one, but the other questions, like asking why that specific phage went for that bacteria,
Mm-hmm.
and it's kind of like the same answer for each, because
Which is?
that phages are specific to like that bacteria. They won't go for another bacteria if they don't match, basically.
Yeah, why?
And so, what do you mean?
It's the question is why each phage is specific. You said because phage are specific is not an answer.
I know.
We ask you. We know that they are specific. But I mean, why?
That is what is like stopped me from turning it in because it's like embarrassed with my
No, you're good, But I mean, the question here clearly, why? Why you think phage are specific? We know that they are specific, but why?
Is this, can I read you my response? And see
yes, yeah, go ahead. Yeah, go ahead.
says, I said plaque appears, which means the surrounding cells are infected and killed by the released virus. A clear spot develops, and I said a positive result is formed from the lysis of the bacterial lawn.
Yeah, this is you're explaining, those plaque formation. Okay, what you are answering here, you're explaining how plaques form, right?
I'm not answering why though, right?
Yeah, why here is specific? Because for each phage, for each phage to attach to a specific bacteria, there should be a specific receptor that the phage recognize.
God.
Okay,
Yeah.
so there's a difference between T4, for example, and I remember I forgot that. Oh, man, Phi X, Phi X174. They are both E. coli, okay, but there is some differences in the cell membrane. So they have some receptors different for the phage. So each phage recognize a specific receptor for attachment. And that is the key here. Okay, so it's very, very specific because the uniqueness of the receptors on the host cell that they recognize, basically.
Well, I mean, I think maybe on question 4, we're basically answering the same thing again.
Yes,
why
exactly.
Did each phage infect
Yes.
bacteria?
Yes.
Yeah, they're all like roundabout ways of answering the same thing.
It will be very interesting if you go and look for what T4 phage, what kind of receptor is recognized by T4 phage, and what kind of receptor is recognized by Phi X174. You will see different kind of glycoproteins and in different location. One of them is recognizing a specific protein in the flagella, for example, flagellal antigen, which is absent in a different bacteria. So this is how specific those agents are. So this is why if you look at this picture here, what happened here? What happened is you have one phage, two different kind of E. coli, like strains, B and C. So why it react with B, not with C? Definitely because bacteria E. coli B and C, they are different in the receptors content that recognized by T4. So T4 recognizes a receptor in E. coli B, which is absent in E. coli C. This is why you end up with no infection here.
That makes a lot of sense.
Yeah, this is mainly that thing about how phage work. Yeah. Okay. But I'm not going to ask you this question. It is not there.
Very, very happy I didn't turn that in yet.
So I'm giving you some hints here.
I would have missed it too.
I like gave the most generic answer, I have to admit.
Yeah, so this is for that lab. Any other lab that you think kind of needs some more from you? Or something for me to explain?
The Hellmann's was pretty straightforward.
Names, you need to recognize, you need to remember, you need to remember those names. Let me go here and see what I have.
Man, by the way, that is some gross stuff.
Yeah.
Yeah, they are, yes. You need to know, the main two phyla that are under Helminthias, and those are nematodes and the platyhelminthias. Okay, you need to know the difference between the two phyla, why they are separated. And then after the below phylum platyhelminthias, there are two classes, trematodes, we call them flukes, and cystodes. You need also to know the difference between them. So if I give you a picture like that and ask you, you know, what is the common name, for example, for this, okay? And what is the general name, for example? If I say, what is the genus of this, okay? The genus here is Fasciola, okay, hepatica, okay? Those are, we call the common name from those are flukes, you know. So you're not going to have so much, but it is mainly memorizing some names, basically, and being able to, for the cystodes, it is important to know some of the features, some feature structures. You need to remember and memorize some names here, okay? So if I give you a picture like that and give you an arrow asking what is the name of this, the head or the scolex, you know, you need to be able to identify this for me, for example. I'm not going to like give you like a life cycle like this and ask you for anything like that. You know, it's not really necessary. But the lab is important in case, or you know, you need to remember and recognize those different kind of worm, and being able to give me the name of the genus if I ask you to do that. Okay. This is for that lab. The other lab, which is kind of, you know, this lab, the immunology and serology, you need to know the difference, you know, between an antigen and antibody. What is the difference between the antigen and antibody? And for serological reaction, you can detect specific microorganism either by looking for the antibodies, or that was basically induced by that particular organism, or by looking at a specific antigen associated with the parasite or the organism. Mainly those are two different ways, okay? If you know this, you know the mechanism, how this thing work, definitely I'm not going to ask you how it works, but it's a very nice schematic diagram showing you how this kind of reaction is happening, okay? You understand this, the details about this particular slide?
We don't see
We don't
anything.
have it. It's not showing up on the screen.
Oh, I'm sorry. I'm so sorry. Let me see what.
No, you're fine.
Okay. So my computer getting older now, and I have some trouble with it.
Okay, this one.
Yeah. So I'm sharing it with you right now.
And this, which handout is this?
This is for the serology and immunology lab.
Okay,
Yeah.
got it.
3 different kind of serological reaction, and you need to know which one was detecting the antibodies, which one was detecting the antigen.
Yeah.
Basically, this is table here.
Mm-hmm.
Okay. My question was here. Do you know what is the basic or the scientific basic behind this? How this thing work? You are basically detecting antibodies here. So you have a virus, and that virus, when it infects, it produces antibodies, and those specific antibodies, we call those heterophilic or heterophile antibodies, which are different from regular antibodies, because the inducing agent here, like the infectious agent or the disease, the virus, the EPV virus, is inducing an immune response, and that immune response resulting in an antibody, but that antibody is not going to be directed to the inducer. It's not going to be directed to the virus, and instead it's going to be directed to a specific other antigen completely different. Is why we call those heterophilic antibodies. In this case, in this specific virus, the antibody that is produced is specific for like antigen on red blood cells, okay? So one way to be able to detect whether an individual is infected with the virus is to look for these antibodies. And the way they do the test here, they know that the antibody react with specific antigen on the red blood cells. What they do, they already extracted or purified those antigens from red blood cells, okay, and they like immobilize it on like this kind of latex here. Okay, so those are not red blood cells, okay. So the antigen from red blood cells is purified. It's a protein which is purified and immobilized on those latex head. And then you add the patient antibodies. You get serum, and you are going to detect if really the person was infected with this virus. If yes, the antibody is going to react with those antigen, and what is going to happen? Those latex particles will get together and will be kind of condensed a little bit and making this kind of appearance here. It's like a precipitation, coagulation testing, basically. Has nothing to do with blood, okay? What blood here? The blood part is the antigen that was extracted from red blood cells, because we know that the antibody can recognize those, okay? So also you need to know this one. Other than that, if you have anything in this lab, you just let me know.
No.
Okay. I was going back through like the very beginning of like the IMVIC testing, like when we first started the bacteria identification.
Yes.
And I had forgot that for the methyl red and of course now I can't remember the other one.
The phenol red.
Yeah, they both like
Yes.
do the stripe
Yes.
of red, or
Yes.
the one turns red. So are the like don't they both use the Kovac Kovac reagent, or is it two different two different kinds of?
Let me try very much to catch up here and see which lab. So, for example, are you talking about the gram-negative Enterobacteriaceae lab, for example?
Yes.
Okay, let me try to grab that one and see here.
I was like watching videos of just how to do the lab, because obviously we're at a disadvantage there, and
Yeah,
you know, he was going through the reagents or what you would add to figure it out. And I just didn't put that together at first, that we're basically using similar. Like,
okay. Can you see this? I'm trying to share it with you. Can you see? Let me here. Yeah, I'm sharing it with you right now. Okay.
yes.
Yeah. Okay. So, which one are you asking for? Which test are you specifically pointing to?
Okay. So yeah, the indole has the red, but I think it's methyl red, or yeah, methyl red, and then the indole test. I think those are the two.
Completely different test. First of all,
yes.
Yeah. First of all, I'm glad that you brought this, because if I give you what is SIM stand for, you need to tell me. We'll ask you this kind of question here. SIM. This is kind of a reaction or biochemical test that can be used in the single tube to identify three different features of the microbe under investigation. So S stands for the hydrogen sulfide production, I stands for indole production, and M stands for motility, okay, testing. So for this particular test here, we don't need to worry about motility. We need just to worry about those two things here: hydrogen sulfide production and indole production in the same media, in the same tube. Okay, so if your organism is capable of using sodium thiosulfate and producing hydrogen sulfide in the media, hydrogen sulfide is colorless, and one way to be able to detect hydrogen sulfide is by adding ferric ammonium sulfate. Okay, So
Okay.
ferric ammonium sulfate, you add this after the reaction, after the growth. It is not included in the media. This is a
Okay.
reagent that you add later and detect hydrogen sulfide. If there is really hydrogen sulfide there, it will react and give you a kind of black color. So here you have two different kind of SIM media. One of them is kind of black and the other one is colorless. The main difference here that this bacteria was capable of producing hydrogen sulfide. This is why it is black. That bacteria couldn't. So this is one test. The same tube, if you are looking for indole production, if the bacteria is capable of converting tryptophan amino acid with an enzyme called tryptophanase, it will degrade or biodegrade this tryptophan and produce indole ring. So one way to detect indole ring is by adding the reagent here, Kovac. So Kovac is a different reagent from this. So Kovac is going to react with indole ring if there is any, and it will form you this very nice, like very reddish cherry color, pink color. Okay, that's an indication of positive for indole. In this here, there is also nothing. Okay, so let's take this to like practical. For that isolate, number one, this is how the test tube SIM look like after adding the reagent.
Yeah.
So first of all, you look here, it is not black. So there is no hydrogen sulfide production. And you look here, there is no pink indole ring, right? And pink color. So there is no indole ring formation as well. So it is negative for both. Okay. The methyl red is different. Methyl red, this is for a different kind of reaction. Okay, and that is the whole thing is IMVIC. Here you are basically looking for glucose fermentation. Okay?
Yeah.
And for glucose fermentation, you are looking for two different kinds of end products. One of them can be easily detected by methyl red. So after you take your organism and you put it in this media, the Vux Proskar media, you incubate, and then you need, it's time now for detection of the fermentation product. You take aliquots from this media into two different tubes, okay? So one of them you add methyl red. Methyl red will detect any acid production. Methyl red in the presence of acidity is going to stay red, okay? It is completely opposite of phenol red. Remember, this is very confusing here for students. Methyl red and phenol red are pH indicators. The color is different completely. Methyl red is positive when it is red. It's acidic. Okay? When it turns yellow, it means that there is no acid. So it is kind of negative, which is opposite in case of phenol red, completely opposite. So if you memorize one test, the other one will be opposite. You don't need to confuse yourself. So I will study methyl red. I know methyl red as a pH indicator. Under acidic condition, it is red. If the condition is basic, it is yellow. So if I ask you about phenol red, you are going to reverse the picture.
Okay.
So this is one thing. The other thing, you are looking for another product from glucose fermentation, which is neutral. It is not acidic. So in this case, you will not be able to use methyl red for detection. Instead, you are going to use those Barrett A and Barrett B reagent for detection. Okay, and you look for this very nice color, red color that is formed.
Okay.
Yeah, that is going to be lots of questions from those kind of testing. You need to remember reagent added, pH indicator, media positive or negative, how they look, how they change color. This is so important.
And the bromthymol blue, it's in the medium, right? Like
yeah, bromthymol blue, yeah, in the Simon citrate media, you have bromthymol blue, which is green under
Right. It's
before.
just we don't add it, right? Like it's
Yeah,
already.
no, yeah, you don't. Yeah, exactly. It's a pH indicator that is going to be in the presence of acid, like formation, like citrate, because you are looking for citrate production here. So change color from this very nice green color to blue color. So that the blue color here is an indication for positive. So if I give you a tube like that and said that tube was inoculated with bacteria A, for example, is this bacteria capable of utilizing citrate or no? Okay, you said no because the color did not change. Then I ask you what name the pH indicator. Then you need to tell me it's bromthymol blue.
Okay.
Yeah. It will be this kind of question. It will be like one line. Most of it will be like one line. You know, some of them one question only. Some of them like 2, 3 together. You know.
Okay.
Yeah.
Are these going to be like our last practical?
Exactly.
I like
Yes.
think we had to fill in most of it. It wasn't like a whole lot of multiple choice questions.
Yes.
Will
Yeah.
this have more multiple choice? Or?
Some will be multiple choice. Some will
Okay.
be fill in the blank, and some will be like a one sentence, like one line answer. You know, don't
Okay.
need to elaborate too much.
Okay.
The bottom line is you need to know the name of the media, the purpose, the indicators, reagents used, all those names you need to be memorized. How they look if it is positive, how they look if it is negative. Okay. Remember here, when you are looking for the decarboxylase test, we have a different pH indicator, and that it is similar here. This is bromthymol blue, that one is bromcresol purple, and this is why it is purple like that. Okay, if there is a positive reaction, it is going to stay purple. If it is negative, it's going to convert to yellow. Okay, this is number one. Number two is when you do this kind of reaction, you need to add the like oil, as you can see here. So when you inoculate your broth, you need to overlay with some mineral oil, because this kind of reaction, when you look for the decarboxylase, it's an enzyme. Exactly, it is very sensitive to oxygen. So you need to have this kind of environment, no oxygen, because if it is oxygen, that will go like through a tricarboxylic acid cycle, and it will be completely off. You need to focus and force the bacteria to produce the like the amino acid decarboxylase, not other enzymes for tricarboxylic acid cycle. And the way you do this by preventing oxygen, and the way you do this by adding oil, like mineral oil. And definitely the mineral oil here should be autoclaved clean. You don't need to introduce any contamination to your culture. Okay, Scott, do you have any question for me?
Nope, just listening.
Okay. Any other labs that you are really interested in going through?
Are we gonna go like? Are you gonna ask any questions about like morphology? Like show a slide and say, like, I don't know what you could even ask.
You mean like cell shape?
Yeah.
Oh, you mean like giving you like a staph or strep or bacilli? No, that
Yes.
was mainly for the first half. Remember when we did staining and a microscopic examination?
Okay,
that
so
was
we're not
at that time.
expect like a street plate.
No. This is mainly for all those biochemical testing that you went through.
Okay.
You know, the morphology of helminthias and ectoparasites. Yes, you need to know those. What is the difference between a flea and a louse? What is the difference between a tick and a mite? You know, mites and ticks. It's kind of like that.
Okay. It's good to know.
Okay. Anything else for the labs?
No.
The lab should
No.
be like straightforward, kind of easy. But as I mentioned, it will be kind of, you know, you really need to study. You know, study. Do your best in studying for that lab practical. You know. Um,
It's mainly just a lot of components to.
yes, I know. I know. This is the main. This is the tricky part about it. That you have different kind of names, different kind of media, different kind of indicators, and a different kind of coloring. You know.
Yeah.
So
And then
sometimes it will be overlapping. Yeah.
yeah.
Okay, so let me see here. So this is for the lab. Um, Again, for the lecture part, as I mentioned, you have 6 chapters to focus on. Okay, and those 2 previously covered chapters you already even tested for, chapter 6 and 10, and the immunology 15, 16, and 24, 25. I encourage you, if you can, like for like as you mentioned, Scott, for immunology, if there is any slide or any specific part, you just email me that, and then I can record the Zoom and post it. You know,
Yeah.
to try to explain it. So maybe that will help.
Yeah, honestly, I don't even know if I am versed enough to ask you a question on. I'm going to go over it again and see if
Yeah,
I
this is
can
what I
process
mentioned.
it a
You
little
guys,
bit
you
more.
have a specific say, you
Yeah,
know what, this kind of slide I didn't get. I don't know what is the difference between this and that.
yeah,
Something like this. I will take those and I record the Zoom and post it for everybody so you can look at it. But as I mentioned, please study for this test. And the chapter 10, chapter 10 is basically talking about the different kind of antibodies and their targets. Okay, so you need from you, let me see here if I can share with you um, this
and you know
lecture.
what? Really, Professor, all these chapters are so relevant to the current times and situation with coronavirus. Everything that we're studying is, um,
Kind of
you hear.
related.
Oh yeah, for sure.
Yeah.
Yeah, you hear Dr. Fauci or
Oh yeah.
Dr. Burks kind of talking. They dumb it down quite a bit, but um,
It will be. I need from you guys some of the life cycles for viruses. I need from you also to focus on, because the final is it is going to be similar. It will be similar to what you had before for other quizzes and tests. Same format, multiple choice, true or false, fill in the space. You might see like few diagrams and figures, and I will ask you to do some labeling. Again, I will provide you with keywords so you can put those keywords, but you need to study for especially the virus the different kind of like viruses and the replication cycle, how they infect, you know, this is important. This,
Professor,
yes.
can I ask before we get too far away, just basic questions like on the lab, how many questions should we expect? And then on the
Okay,
exam?
the main thing is the lab is going to be all together around 50, okay?
Okay.
So, but what you are going to see in your practical, you are going to see maybe 34.
Yeah.
So why 34? Because some of the question will have multiple,
Multiple
you know what I
parts.
mean?
Yeah.
So what kind of media, what kind of reagent, how it looks if it is positive, like this. So I'm
Yeah.
counting this everyone as one question, okay?
Okay.
So you look at it, you will be maybe around 30 something, but in reality, there will be around 50 something question.
Yeah. Hmm.
Yeah. Yeah.
Okay.
Okay, so for this here, you are talking about, you know, different kind of antibiotics and, you know, the structure of the microbe that can be targeted by those, I need from you to memorize those kind of antibiotics for me. Okay. So if I ask you like what kind of antibiotics you used, say for that interfere with cell wall formation, for example, or inhibiting cell wall, you have plenty of antibiotics here. I need for you to memorize this slide. Okay. So if I ask you, for example, penicillin, how penicillin work, if I give you a diagram or if I give you multiple choice. So you need to know that penicillin is basically an antibiotic that interfere with the cell wall synthesis. It has nothing to do with inhibition of protein synthesis or interfering with DNA replication, for example. Okay. So this slide is important. Study this slide. Okay. I already started, putting together the final. I didn't finish it yet because I have another final for another class that I need also to prepare. So it will keep me busy for a couple of days to finish those. But this is one of the things that I'm really interested to add to your final. Okay. So as I mentioned, to make things more easy for you and more productive, you can email me anything for those 4 chapters, 15, 16, 24, 25, that you was not able to understand or comprehend quite well, due to lack of some information. And I will be more than happy to put together like a recording for to discuss that particular question or concern.
Sure.
Okay, anything else for me?
No.
No. Thanks for.
No problem. I just need from you to study. Everybody's doing great so far, and now it is time for selection.
Yeah.
So that will be tricky. So those 20%, will be kind of hard a little bit. So work hard, work your best.
Yeah, I was like hoping week on, like off weeks, not like a day after each other. So now.
Yeah, unfortunately, because as I mentioned, the summer schedule is so tight.
Oh, I know.
And as you know, every week there are like 4 or 5 or 3, 4, 5 labs, per week.
Yeah.
And normally we have like 1 or 2 labs per week during the fall or spring semester. So it's very condensed. Yeah, I know. So everything is highly condensed. You are heroes, guys. You are really heroes. And you are doing very good jobs so far. And I hope you will continue this. As I mentioned, a few more days, just try to put more energy and do your best so you can successfully, pass this with a good grade.
Okay. Well, thank you so much.
No problem. Good luck. Good luck for everyone. Okay.
Yep. And if we don't talk to you, I guess this might be the last Zoom. Thank you for the semester. And
no problem. I wish I could do
sometime.
better and do more, but this is unfortunately, the situation was like, as you know, it is kind of teaching online is not easy. It is very, very time consuming. But we all went through this and we started it together, and hopefully we'll be able to finish it together in a good way.
Sure.
Well, if I see you on campus, I'll introduce myself and say
definitely
hello.
I will be more than happy. And I will be on campus, as I mentioned. They asked us to be there like very close to the end of the fall semester, around maybe week 10 or so. So we'll see. Yes.
All right.
Okay. Thank you very much. Good luck, guys. Good luck.
Thank you.
You have a wonderful night.
You too.
Okay.
Bye-bye.
Bye. 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 Bye.